So let's talk about transcription, or better RNA transcription. And the purpose of transcription is to make messenger RNA. And what do you need the mRNA, the messenger RNA, for? You need to make it to make a protein. So it's essentially the first step of making a protein. And what you need for transcription, you need an original DNA template to copy from. You need an enzyme called the RNA polymerase, and this RNA polymerase reads the original DNA template, and it basically makes the mRNA. And then, of course, you also need the building blocks to make the mRNA, and these are the so-called RNA nucleotides. And there is no more picture here, but I'm just simply decided to actually explain the whole process using a drawing. And I'm now going to draw my DNA molecule over here. And I'm going to open up the DNA molecule over here, and I'm going to close it down here again. So that's only one strand, so I need to draw the second one over here as well. So that's supposed to be the double helix. Here, it's the DNA is open, and over here, okay. And of course, between here you have the hydrogen bonds, the bases, and so on. So that is my DNA template. That's the DNA template. Okay, and yeah, that's this one over here. And you can already see that essentially the DNA was opened here in the middle. It's a little bit like a broken zipper, which you open in the middle, where you're separating the zipper. And you need to do that. You need to break these hydrogen bonds in order to expose the bases. So, and the RNA polymerase does that. And I don't know, I'm going to draw this. I'm just going to draw the RNA polymerase all over here. It's a schematic drawing, okay? So I call this the RNA polymerase. Okay, that is my RNA polymerase. It's an enzyme. And on the DNA, of course, I have my bases here. And let's say that I have an A over here. I'm just going to invent something here. An A over here, then of course on the complementary bit one over here must be a T. If I have a G over here, there must be a C. If I have an A over here, there must be a T. And then let's say another G and a C. You get the idea, okay? And basically what the RNA polymerase does is the following. It sees that there is a G over here, and it inserts a C. And it has already done this, and over here it has already made mRNA. So I'm going to draw over here this mRNA, and this mRNA now leaves the DNA. That's my mRNA, the one that the polymerase, the DNA RNA polymerase made. In between here, of course, I have my hydrogen bonds, and the RNA polymerase now moves into this direction over there. And the DNA over here in the back, it starts to close again. So I'm going to just draw arrows here to kind of illustrate that it's the DNA strands close. DNA closes again. Yeah, and here over here the DNA, of course, the RNA polymerase opens the DNA, it unwinds the DNA, so to say. So it basically does both. It unwinds it and opens it, and also adds the complementary bases. And now let's have a look. I did not draw the nucleotides yet. So I have, of course, an A. These are the RNA A nucleotides, this little thing here. Then over here I have a G nucleotide, a C nucleotide. And if you think it's a T, then you're wrong. RNA has U. Okay, it's a uracil over here. And there are many of them floating around. Okay, another G over here, another C. And over here, the polymerase now does the following. It sees, there's a T over here on the DNA. This T, what is the complementary for T? It is A. So it will take this A over here, and it will insert it over here. And then you have attached over here an A. And then it moves on. It says here, there's a G over here. It will take the C from over here, and it will attach the C. And it will keep on doing that, okay? And over here, there's an A, it reads an A, so it will take the U. Yeah. And then, until it reaches the end, how it knows when it's at the end, it's a different issue. That's not relevant right now. But at the end of the gene, let's say all the way up here somewhere, yeah, it will stop at the end of the gene, and the mRNA will then completely have left the DNA, and then you have at the very end, you have your original DNA again, okay, which is now closed. That's the DNA, and plus DNA, and plus you will also have your mRNA. And that's it. That's essentially transcription. Just one thing which is important is not the whole DNA is copied, but only one gene of the DNA. It's a relatively short section is copied. In replication, DNA replication, when we actually copy the whole DNA because we would like to have 2 DNA copies. Here we do not want to copy the DNA. We don't want it. Let me correct this. I do not want to have 2 DNA copies, but I would like to have a part of the DNA copied to be made to make mRNA. And some students, or many students, or some students are a little bit confused because there is a similarity between DNA replication, the copying of DNA, and the RNA transcription, where you're essentially also reading the bases. But the point is a different one. Here you want to make mRNA, and because this mRNA contains now the information to make a protein. So the point between transcription and replication are quite different. The purposes are quite different. But you also see that there's this complementary base pairing going on in both. And another thing is that in transcription over here, we only read one strand of the DNA, not the second one here at the top as well. It's only one strand which is read off to make mRNA. Okay, so that was basically that's transcription of that's RNA transcription.